So the focus of my talk is going to be on a publication that was published by the National Academies of Sciences, Engineering, and Medicine in March of 2023. It's a consensus study report entitled Using Population Descriptors in Genetics and Genomics Research, a New Framework for an Evolving Field. Now, before we embark on this talk, I'm going to open up the chat, and I'm going to ask you to put a 1 in the chat if you had heard about this publication before this week. So before you got an email saying, hey, Dr. Deyrup will be talking about this. So if that is the case, please put a 1 in the chat. Once you're working on that, my second question will be to put the number 2 in the chat if you have actually read this publication. So it is a big chunk of change. It's 215 pages if you buy the book version. So if you've read about half of it, please put a 2 in the chat. And then finally, if you are someone who does genetics and genomics research, or who collaborates with someone who does genetics and genomics research, so perhaps you review slides or you're part of the tissue biorepository group, put a 3. But also, if as part of the way that you are staying up with your field, you read scientific articles that address genetics and genomics. So if any of those is true, please put a 3 in the chat. And I see, sadly, the chat is completely bereft of entries. So that tells me that this is a very important topic for us to discuss. So over the next 45 minutes or so, I'm going to review the history of population descriptors. I'm going to focus on this NASM publication, and I'll finish up by talking about how we as pathologists can contribute to accurate biomedical research. So why is this so important? So research in genetics and genomics is evolving incredibly quickly. There are papers being published by the hour looking at genetics and genomics. Unfortunately, many of these studies are continuing to use race-based categories. And this calls into question the scientific accuracy of the entire field because biological race does not exist in modern humans. So before we embark on the NASM publication, let's think about where it all started. How did we start getting the population descriptors we use today in the United States? So we're going to go back in time before the Civil Rights Act of 1964 in the United States. At that time, we had Jim Crow segregation, which was separate and unequal, and which affected transportation, hospitals, restaurants, schools, etc. We also had redlining, so the Federal Housing Administration would not insure mortgages in neighborhoods they deemed risky. And this was predominantly areas with African American groups, also immigrants and low income. Federally subsidized housing was segregated by race. And the GI Bill, which provided excellent benefits to our veterans, excluded African Americans from low-interest mortgages and education benefits. And of course, there were health disparities. So this figure shows life expectancy at birth. The population descriptors they are using are all races in blue, white is in gray, and black is in orange. And this graph comes with a caveat that data on life expectancy in African Americans before 1969 is questionable. So even though these data might not be entirely accurate, you can get an idea of the trends, and the trend is in fact appalling. Now, in 1964, we got the Civil Rights Act, and this was the prohibition of discrimination due to race, color, religion, sex, and national origin. And there are many components to this act. The one that's most relevant to us in healthcare is Title VI, non-discrimination in federally assisted programs, because Medicare and Medicaid are such a strong part of U.S. healthcare. Now, after the Civil Rights Act, there were some additional acts that were passed to shore up areas that needed more attention or areas that had not been addressed at all. We had the Voting Rights Act of '65, the Fair Housing Act of '68, and the Home Mortgage Disclosure Act of '75. So now we're trying to address disparities, but we have to have some way to track disparities and hold people accountable. So with that in mind, in 1977, the Office of Management and Budget, or OMB, issued their Statistical Directive 15. This is the official system of ethno-racial classification used by the federal agencies to standardize record keeping, collection, and presentation of data. So this is an ethno-racial classification. So let's pause for a moment and reflect, what is race? So in modern biology, races are defined by two criteria: the amount of genetic variation within and between groups, and phylogenetic evidence of an evolutionarily unique lineage. And neither of these conditions is met in modern humans. Now, it has long been known that humans show more variability within so-called races than between them. Now, obviously, we have geographically based genetic and physical differences. You need only look around the Zoom room to see that we have these differences. However, we share 99.9% of our DNA. Now, while biological race does not exist in modern humans, socially constructed or socially defined race does. And in fact, that is exactly what OMB Statistical Directive 15 is doing. It is defining race. Now, they're not the first group that has done this. We have seen socially defined race for centuries. And this is the arbitrary use of aspects of morphology, geography, culture, etc. And as it was initially used back in the 18th century, it was in the service of a social dominance hierarchy. And I'll expand more on that in a moment. These are the classifications from the 77 OMB Statistical Directive. Now, in other talks that I've given, I've worked with Dr. Graves to tear these apart. I'm not going to do that in this talk. We're simply going to take a look at them. So we have American Indian or Alaska Native. We have Asian or Pacific Islander. This was split into two groups in the 1997 OMB revision. We have Black, we have White, and we have Hispanic. Now, Hispanic is an ethnicity. It is not considered to be a socially defined race. And so this information is collected separately. So if we look at what we have after 1997, you can see here the 5 races. These are the ones that are used by the federal government for the census. They are used by the NIH, and we see this in the SEER data as well. Now, these look very familiar. As I said, they're not brand new. Here we have a figure from 1795, where you can see these exact 5 races again. And this is at a time in the 18th century when we had extensive colonialization. We had the enslavement of individuals. And by setting up a hierarchy, it was easier for the dominant group to enslave, exploit, torture, and murder these other individuals because they were considered to be less than. And this is a legacy that maintains to this day. Now, going back to our history, in 1985, we had the creation of 2 task forces by the Department of Health and Human Services. The first one here is the one on Black and minority health, and one of its central goals was better data collection, so that, for example, those life expectancy tables could be accurate, because it was important to compare across these different groups to address these disparities. The other group was focused on women's health issues, and part of their mandate was to ensure emphasis on conditions and diseases unique to or more prevalent in women in all age groups. And this was really driven by the strong feminist movement by women's groups. But one thing that both of these groups also addressed was clinical trials. There was a problem with clinical trials in the United States. Nearly all study subjects were European American men. Now, this was in part because they were easier to recruit. If you needed a lot of subjects, you could go to a military base and there would be abundant people you could recruit. If you needed a specific hospital population, you could go to a VA hospital. So they were easy to find. They were also easier to assess. They didn't have monthly hormonal fluctuations, and they were less risky. You didn't have to worry about your subject becoming pregnant or the risk of birth defects if you were trying a medication. Now, this was obviously to us now very problematic, and it was obvious to some people back then, including Dr. Vivian Pinn, who was the president of the National Medical Association from 1989 to 90. Dr. Pinn will be giving grand rounds for Duke Pathology next week, so we're very delighted and honored that we will have her presence here. But what she said in 1990 is that some of our physicians are a little leery of some drugs because we can't be certain whether minorities have been participants in clinical trials. Now, this quote comes from an article in the Journal of the American Medical Association with a title that I think sums it all up. Is there still too much extrapolation from data on middle-aged white men? So this began the process of the NIH Revitalization Act. Now, there was an initial draft in 1990. This is described very well by Dorothy Roberts in her book, Fatal Invention. And again, because of the advocacy of women's groups, it was mandated that females, be included as research subjects. Now, when the Congressional Black Caucus sees this 1990 draft, they also lobby very hard. We also have to have minorities. It cannot simply be European Americans in clinical trials. But this is part of the origin of the problems that plague us today. We have a biological category, so sex. We know that females and males have differences in biology, and we are adding in a socially defined category. And the problem with this is that it opens the door to conflating a biological category with a socially defined category. Here is that NIH Revitalization Act of 1993 showing that we need to include women and minority groups. The NASM publication is very explicit here. The fundamentally socio-political origins of these racial categories make them a poor fit for capturing human biological diversity. Now, the way that we do it in the United States is not the way it's done everywhere. This is a table that comes from the NASM publication that is looking at 3 distinct global contexts and the genetic studies that are done using them. So on the left, we have the UK Biobank, which divides people into white, mixed, Asian, Black, Chinese, and a couple of others. By contrast, if we look at the Health and Aging in Africa study, they're dividing people and classifying them by native language because of perhaps the belief that this would be a more discrete population because there'd be less mixing between language groups. And then if we look at Brazil, at the Brazilian Initiative on Precision Medicine, they are purely using geography. So as you can see, there are lots of different ways to categorize individuals, and this should raise the problem for you that if you're doing research, how are you going to compare data from different parts of the globe if we're all using different population descriptors? And the NASM publication will provide guidance on that. So in order to demonstrate for you the challenges of population descriptors, let's begin by looking at a case. So for those of us who are attendings in pathology, if we choose to, every quarter we can get a package from the College of American Pathologists Performance Improved Program. This gives us 10 cases with virtual or glass slides. There's a clinical vignette. There are multiple choice questions, and there's a lengthy discussion for each case. This quote comes from a case from late in 2022. And what it is addressing is an amyloidogenic allele that they describe as a hitherto under-recognized cause of cardiomyopathy in individuals of African and Hispanic ancestry. Now, this sentence from the CAP-PIP discussion comes pretty close to the actual title of the article they cited. So this is an article in the Journal of the American Medical Association from 2019 that you can see here. Now, before we look at that article, let's reflect for a moment on this actual disease, which is hereditary transthyretin amyloid cardiomyopathy. So you get this disease when you have a mutation in the gene that encodes transthyretin, that among other functions transports thyroxine. If you have a valine to alanine substitution at position 122, you have a 4-fold increase in your risk of amyloid formation. And these fibrils can deposit in a variety of tissues, including the nerves and the heart. It is a cardiomyopathy that leads to death about 2 to 6 years after diagnosis. Now, this allele is autosomal dominant, but it has variable penetrance, which means that just because you have this mutation does not mean you get the disease. Individuals who are affected tend to be males over the age of 60. Now, patients can present with signs and symptoms suggestive of hypertensive cardiomyopathy, but they are typically normotensive. Epidemiologically, we know that about 3 to 4% of African Americans have this allele, compared to about 0.4% of European Americans. Now, back in 2016, it was recognized that this allele was seen in some individuals with African ancestry, and Jacobson and colleagues looked at the prevalence and distribution of this allele in Africa. And you can see their data here, where this dark orange area is where you see a higher prevalence. And their conclusion was that this was probably a de novo mutation that arose in Western Africa and then spread from there in a somewhat centripetal fashion. Now let's turn to the 2019 JAMA paper that is cited by CAP. Now, this is a complex paper with many different components. I'm going to focus on one component at this point in the talk. So they took about 42,000 participants of a biobank that is housed at Icahn School of Medicine at Mount Sinai in New York City. They were able to get high-quality whole exome sequencing on about 27,000 of those people, and they were then looking to identify whether or not they had the allele. Now, this biobank is tied to the electronic health record, so they used that to find the self-reported ancestry or country of origin for these individuals. This is one of the figures using those data. Now, let me show you how we're going to interpret this. So the closer the color comes to red or orange, the higher the carrier percentage. The size of the circle lets you know what the sample size was. Now, many of these samples are quite small, less than 50, often just a handful of cases. And that is because these are patients living in New York City who are reporting their country of origin. Now, when we look at this image, it looks very similar to what we saw in that 2016 paper, where we have an increased prevalence here in Western Africa. But things get much more complicated when we look at this part of the globe. So what we see here, first of all, is that the circles are much larger, which makes sense because the biobank is in New York City. But we also see a surprising prevalence of this allele in North, Central, and South America. Now, the title alludes to Hispanics, but if we look over at Spain, we see that there doesn't seem to be any of this here. So is this perhaps due to some new mutation here on our part of the world that accounts for this incredible prevalence? No, that's not what is thought. So in order to understand this, we look at genetic ancestry of Latin American populations. So what is done to do this is you take your study participants, you do genetic analysis, and you compare that to a reference population. The reference populations they used here, they classified as Amerindian, European, and Sub-Saharan Africa. Now, if we look here at Puerto Rico, you can see that about 12% of the ancestry comes from Sub-Saharan Africa, and that makes sense. We know this was an area of sugar plantations, and many enslaved Africans were brought to this part of the world. Now, as you look at the other countries that are examined, you can see that the sub-Saharan component is much smaller, 2% in Peru, 3% in Mexico, 7% in Colombia. So you may be thinking, well, it's a very small component. This is a rare allele. What's going on here? Now, remember, sampling is extremely important. So let's just focus on Colombia. This is a map from Wikipedia showing the country of Colombia and two study areas from a 2017 publication. And what they did was they compared the urban center of Medellin to a region, one of the states of Colombia called Chocó. Now remember that the country as a whole is 64% European, 29% Amerindian, and 7% Sub-Saharan African. And when they did their analysis here, these are the results they found. Now, let me explain how it is that we interpret these figures. So each of these lines is an individual. So if we look, for example, at this figure here in Chocó, we would say this individual has perhaps 10% Sub-Saharan African ancestry, maybe 60% European, and the rest is Native American. And you can see here that it's quite different between what we see in Medellin and Chocó, with Chocó being about 76% Sub-Saharan African ancestry. The reason for this is that this state was a site of gold mining. And so in the history of Colombia, many enslaved Africans were brought here, and this is the ancestry that we see in these individuals now. Now, one of the things that they're using here by using terms like African, European, and Native American is something we will refer to as continental ancestry, and we'll address this in the NASM publication. Now, as we look at the United States, we see a similar admixture in Latin Americans in the US. So again, we have a tri-admixture with African ancestry in blue in the upper left, mean European ancestry in red on the right, and then Native American industry on the lower left. And you can appreciate immediately that it is regionally variable. So if we're looking at the western half of the country, there's a very small component of African ancestry compared to the eastern half, particularly when we look, for example, at New York City, which is where this biobank was housed for the 2019 study. Now, the authors of that 2019 study were aware of this. In their discussion, they state explicitly that the individuals of self-reported Hispanic and Latino ancestry from the New York City region are known to have significant levels of recent African ancestry due to admixture. So this would account for a higher risk of this allele. On the other hand, if we look at West Coast Hispanics, we would expect this to be extremely low. So the authors did understand this. But what we see when we are looking at this paper is this title. We look at the conclusions in the abstract, and that is what we get. And that is the information that is disseminated through the College of American Pathologists. So many of us are too busy to go through the detailed workings of a paper. This particular sort of idea I consider to be clickbait, because many people are interested in demonstrating genetic differences between the different races. And so this is what we are sharing with the world. Now, at the end of this talk, I'll come back to it and give some pointers on how this paper could have been better. Now, I was showing you some images looking at continental ancestry across different groups. Keep in mind that, again, 99.9% of our DNA is shared across modern humans. That's a 0.1% difference. That's about 3 million base pairs, or 1 in every 1,000 base pairs is different between individuals. This is a very important quotation from the New England Journal of Medicine from 2003, because people might say, well, that's where the difference is. Those 3 million base pairs, that is where race can be found. What Cooper and colleagues say is that genetic variation is continuous and discordant with race. Systemic variation according to continent is very limited, and there's no evidence that the units of interest for medical genetics correspond to what we call races. So how can the NASM publication help us in what we are doing? So let's begin by talking about what is in the box. So this is a significant publication. You can download the PDF for free. I'm a book reader, so I have the book, 215 pages. It's 9 boxes, 5 figures, 5 tables, and an excellent glossary. There are conclusions, best practices, recommendations, and detailed decision trees for various study types. This is not, however, an improved nomenclature or vocabulary. It's not simply where you used to say the word Black, now say African American or African descent. It is not a cut and paste pattern for the replacement of problematic terms. What it is a challenge to the misconceptions that are rife not only in medical research, but in healthcare. It is an invitation to open the dialogue further as more of us learn the information that is here. And it is an approach to an evolving discussion, because this field, as I mentioned, is changing rapidly. This figure provides a roadmap of the NASM publication. So on the left, we have the principles that guided their decision when they came up with the population descriptors. So these include the ethical principles of respect, beneficence, equity, and justice. Now, I personally know researchers who say to me, that is a woke agenda. I am a hard-nosed scientist. I care about hard data. This is soft. Well, if that is your position, you should care about these additional scientific principles of validity and reproducibility, transparency and replicability. Because if your science is not valid or reproducible, if it's not transparent and cannot be replicated, then it is bad science. Using those principles, the NASM group came up with the requisites that they thought were essential in order to come up with appropriate population descriptors. These include avoid typological thinking, which I will define shortly, measuring environmental factors, don't just use race or ethnicity as a proxy, and engaging communities and participants. They then give guidance for researchers. They come up with 5 different categories of research studies, and I'll talk about that later. And in addition, they talk about the importance of implementation. The goal is not to make an excellent paper that then sits on the shelf. It is critical that we involve stakeholders such as study participants, funders, journalists, professional societies, journalists and media. And I'm quite pleased that Annette Flanagan, who is the executive managing director of the Journal of American Medical Association, is on this meeting today. Now, in my talk, I will be focusing particularly on these issues, beginning with avoiding typological thinking. So what is typological thinking? It's when you classify individuals as set types. Now, this ignores variation among individuals within the group. So, for example, it is commonly said in the United States, African Americans, they're at increased risk for sickle cell disease. Well, yes, some individuals of African descent are at increased risk for sickle cell disease, but there are parts of Africa that do not have a history of malaria, do not have a prevalence of the hemoglobin S allele, and individuals from there, whether they came here many generations ago or are recent immigrants, are at no increased risk for that disease. And the problem with using types such as this is it can reinforce prejudices about these groups, because remember, these racial groups have a long-standing history in a social dominance hierarchy. Now, for those of you who think that we are in a post-racial world and that we are all beyond this, I highly recommend the book Biased by Jennifer Eberhardt. It was incredibly eye-opening for me. So the NASM publication has the following conclusions regarding typological thinking. It's quite simply, terms that imply a biological classification of race should not be used because race is neither useful nor scientifically valid when we're looking at the structure of human genetic variation. Now, socially defined race is very important for socially defined problems, so public health, access to care, vaccination, etc. They emphasize that using race as a variable can be harmful because it suggests that the health disparities that we are seeing are due to innate biological differences. This can cause us to pay less attention to root causes, such as toxin exposure or lack of access to care. And they also state quite clearly that the current practices being used in genetics and genomics research reinforce typological views of human genetic ancestry. For example, the use of continental ancestry groups, which I referred to in that paper about Chacot and Medellin, and I'll talk about that more in a bit. So they say we need a better way to classify people, a different category, and they recommend genetic similarity, which I will also talk about in this presentation. Now, they have two very important points about typological thinking. The first is that using these OMB categories, the very categories that the NIH says to you, you must use to report your participant demographics, perpetuates misconceptions and exacerbates typological thinking. Now, they give us a way to address this. So they say it is important to use our OMB to satisfy the reporting purposes, but you don't need to use these typological categories in your analysis. So yes, we want to make sure that our study participants are not all European American males. We do want to document 20% African American, 20% Asian American. But when you get your analysis, if you use these racial groups, you will find statistically significant differences between these socially defined races. And that is because the impact, the enduring impact of systemic racism. You'll get something statistically significant. You won't be able to resist publishing that. And then this adds to the mountain of literature that supports race-based medicine. Next, I will talk about measuring environmental factors. So these are the physical, chemical, and biological exposures that subjects can have. And it includes the social context. So that we know, for example, systemic racism with the discrimination and bias that individuals experience can lead to changes to their epigenetics. We have increased release of stress hormones. This has been referred to as weathering. These environmental factors may act independently on phenotype, or they can interact with the genome, and they may do so either in an additive fashion or multiplicatively. Now, environmental factors can be very difficult to measure. We used to think, say 30 years ago, we were doing a good job if we looked at educational attainment or household income or rural versus urban. There are so many environmental factors that we're not even aware of, that we don't even know to measure. This is one of the reasons that race and ethnicity are frequently used as proxies. This is a paper that came out in 2022 that's looking at present-day air pollution disparities in US cities and aligns that with historical redlining. So what they are noting is the presence of fine and ultrafine particles in the air, and it is more common in areas that have been redlined. Why is this? Well, if you have an urban area that the FHA will not provide insurance for mortgages there, you get urban decline. And when you have urban decline in these areas where you had African Americans or immigrants, then we would get industry there. So factories and foundries there. And this in part contributes to the present day presence of these increased particulate matter in these areas. Now, we didn't even know about these 30 years ago. We're only looking at it now. And in her book, Fatal Invention, Dorothy Roberts talks about a number of researchers who remain determined to show that there is a genetic difference that predisposes Latin Americans and African Americans in public housing to asthma, even being aware of these prevalent environmental factors. This is something for Dr. Howell, because as we are learning, these particulates, you would think they would obviously have an impact on the lungs. They can also have an impact on other organs, such as the kidneys. Now, we make a big deal about trypanosomes and ApoL1 when we talk about chronic kidney disease in African Americans. But now, as we look at this and we recognize increased exposure to particulate matter that also contributes to kidney disease, you see that we are looking for a biological or genetic reason when it is actually a social environmental reason. So the conclusions in the NASM publication are that you must directly evaluate the relevant environmental factors or exposures. Do not simply rely on population descriptors as proxies. Now, they recognize it's not always possible. So say you measure everything you can, you still see differences. You can use population descriptors such as race and ethnicity if you explain how they are used, why they are used, and why they are relevant. And in order to do this in the best possible way, they recommend collaborating with experts, for example, in the social sciences, epidemiology, environmental sciences, etc. Now, this may cause a little bit of pushback because this is a little bit more work. We know how to find other pathologists or other clinicians. How am I going to find a social scientist or an epidemiologist? This is too much work. But if you think about it, we do this on a weekly basis at our tumor boards, our multidisciplinary conferences. In those conferences, they are quite diverse. We will have a surgeon, a pathologist, a medical oncologist, a radiation oncologist, a radiologist. And we do this team-like approach for optimal patient care. We should also keep this in mind for optimal research. This brings me to the portion on the guidance for researchers. So the NASM publication divides genomic studies into 5 groups. We have gene discovery, trait prediction, cellular and physiological mechanisms, health disparities, and human evolutionary history. Now, when we're talking about population descriptors such as race or ethnicity, those are large baskets. And we would anticipate that everyone would have similar cellular and physiological mechanisms. We may see differences in small inbred populations, but in general, this is not going to be those large European ancestry group, or sorry, continental ancestry groups. So we can remove that for right now. Now, we in general don't focus on human evolutionary history, so we can get rid of that one too. So that gives us 3 study types. The NASM publication breaks gene discovery and trait prediction into 2 groups, whether they are Mendelian or complex traits. So we will end up with 5 genomics study types. Now, the NASM publication will give us a variety of different population descriptors to consider. We have race, we have ethnicity and indigenicity, geography, genetic ancestry, and genetic similarity. And here is a beautiful figure that lets you know what it is you should use for your study type. And this is the key. So red should not be used, and you'll see the area where that really shows up is race. Yellow with a question mark means in some cases you can use this population descriptor, And yellow with an E means it can be used if it's an appropriate proxy for environmental but not genetic effects. And then green is our preferred population descriptors. So you can see that race is really not useful at all unless we are looking at health disparities with genomic data, and you have to emphasize it is a proxy for environmental effects. Now, clearly the gold standard is going to be genetic similarity. So what is genetic similarity? This is a quantitative measure of genetic variation between samples. You can think of this as genetic relatedness. And the way that you come up with this is you will take an individual genome and you will compare it to a reference population. Now, you have to keep in mind that you have to choose your reference population carefully and not use biased reference samples. So, for example, if you're doing genome-wide association screening, this gets very challenging. So according to GWAS-DiversityMonitor.com, about 95% of total GWAS participants are European. This makes it very difficult if you are studying other populations, and again brings up this idea of the universal norm. So there are a variety of different ways you can measure genetic similarity. These are statistical programs. One of them is called admixture. And what this will do is it will define groups with similar patterns of variation, and it will yield something called an admixture fraction. And we have already looked at one example of this when we talked about Medellin and Chocó. And so what we see in their analysis is that the subjects from Medellin had about 75% European ancestry when compared to that reference group, and subjects from Chocó had about 76% African ancestry. But this immediately raises that problem I mentioned of the continental ancestry group. So perhaps in the study, they used the Yoruba as a reference population. The Yoruba are individuals who are in the western part of Africa, and they are frequently used as a reference population for sub-Saharan Africa. So when you go to the trouble of using a specific population like that, but then you just revert back to the continent, African, European, Native American, these are 3 of the 5 races that we use in the OMB. So we are right back in our typological thinking. The NASM publication will help us with this. Now, another way we can use is principal component analysis, which is a 2-dimensional plot that shows correlation across variables. And here is another example from that same paper. What you can see here is that the individuals from Medellin are going to cluster with the European reference populations, and the individuals from Chocó are going to cluster with African reference populations. In fact, more specifically, they're going to cluster with West African reference populations, the Yoruba and the Mende. Now, as I've referenced before, we know that African Americans tend to have more West African and Central African ancestry because that is a region from which enslaved people originated. Now, if you're looking at this graph, particularly if you look at flow cytometry, you might be thinking, wow, these populations are really different. Keep in mind, we are looking at 0.1% of the genome. So if you're thinking about perhaps a flow cytometry graph and you're looking just at 2 markers, yes, you can differentiate those 2 populations, but there are so many other proteins that are on those cells that are identical. So that might help you as you think about principal component analysis here. So to sum up, genetic similarity is a quantitative measure of the genetic resemblance between individuals that reflects the extent of shared genetic ancestry. And one reason I'm emphasizing this is because if it is so important in genetic and genomic studies that we do this right, we should bring the same rigorousness of thought to what we do with the rest of our studies in pathology. So the recommendations of the NASM publication are: first, use well-recognized reference sets. So they mentioned the 1000 Genomes Project, which has diversity as part of its goal, and the Human Genome Diversity Project. And they say, when you report your population descriptors, please be mindful. Don't use the Yoruba and the Mende as your reference population and then revert to race, ethnicity, or continental geography, because that throws us right back into typological thinking. They recommend that you use terms that reflect the complexity of the problem as opposed to oversimplifying the issue. And they recommend using the suffix -like. So what does that look like? So here is a sentence that you might see in a genetics or genomics paper, in which they are trying to have a homogeneous population for their analysis. So what they say is to minimize that heterogeneity, they're going to use a pairwise genotypic dissimilarity, less than 10 to the minus 3. So in other words, they're trying for a homogeneous group. Remember, 1 in 1,000 is the base pair difference between individuals in general. And they want these individuals to be very close to a particular sample, so the mean of the Yoruba of Ibadan sample in the 1,000 Genomes Project. Now, that is a very lengthy description, and if you had to use that throughout your paper, it would really chew up a lot of your paper, and it's not easy to follow. So they recommend using a sample abbreviation and the suffix -like. So for this particular example, you would use 1 kg for 1,000 Genomes Project, YRI for Yoruba of Ibadan, like individuals. Now they acknowledge it's a little vague, but it is concise, so you can use that throughout your paper. You will elaborate the meaning in the methods, which you see here, and then your abbreviation will be sprinkled throughout your paper. And when the reader sees that, there's going to cause a little bit of discomfort. What does this mean? They will then look into the methods. Now, if you just revert back to saying, well, the Yoruba for Western Africa, we'll just call a continental ancestry group, say they're African, there is no need for that reader to go back and understand what you're truly doing. Now, this brings me to the final part of my talk. What can we do as pathologists? So first, please read this publication. It is an extraordinary resource with checklists, decision trees, and specific guidance. Please urge your research colleagues, the ones you're collaborating with on these genetics and genomics studies, to read this publication as well. There's a lot of information, and it's going to take all of us to make sure this is implemented. And remember, as you're analyzing your data, that just because you need to tell the NIH what your recruitment is, you do not need to use that as your analytical tool in your research. This is just an example of one of those decision trees to give you an idea of how it works. So it begins with the question, what is the purpose of your study? Here it is gene discovery. It then takes you down, is the trait complex or Mendelian? And then you go down through the decision tree, and you can see here we have our key again, saying, yes, you should use genetic similarity. No, do not use genetic ancestry. So it's useful to use these trees as you are conceiving your project, as you are writing your NIH grant, and as you are writing your paper. They also recommend that you ask questions. I'm not going to read through these because these are actually in the NASM publication. These are questions for authors. But I also think they're ones that all of us can use when we are reviewing papers. So if you are reviewing a paper and you see population descriptors that are either based on race or ethnicity, use these questions to poke the authors to make sure that their report is accurate. So I'm going to finish up with how could this study be better? So first of all, don't use race or ethnic labels. So if we look at this figure here, we could say, yes, we have an increased prevalence of this allele in these 6 countries. Or we could say in Western Africa, but to say everyone of African ancestry is clearly inaccurate. You can see the vast areas of Africa that do not have this allele. Furthermore, we know there's more genetic diversity in Africa than anywhere else. So this sentence is quite frankly false. One aspect of this paper that I did not go into was they had 2 components to this. So the part that I showed you was based on self-report from the electronic health record for people to say what their self-reported ancestry or country of origin was. For another portion of the study, they actually did do genetic analysis, where they took a study population, they compared them to a reference population, and they had individuals of inferred African ancestry. And you can see this is 2 arms of that study. Now, if they had done this genetic analysis on both arms of their study, they would have recognized that of the self-reported Hispanic Latino ancestry, they all had an African contribution. Finally, please be careful with your titles. As I said before, this title is clickbait. There are many people in this country who are very interested in race-based medicine and any evidence they can find that supports racializing medicine. So what does the future hold? Well, we're not going to go that far. Let's talk about what happens in March. We're quite fortunate that one of the co-chairs of the NASM publication is right here at Duke, Dr. Charmaine Royal. And she and the other co-chair will be presenting on Tuesday, March 19th, looking at this publication and what has happened since it was released in March of 2023. Dr. Graves and myself will be at USCAP this year for the American Society for Clinical Pathology Companion Society for a talk that will include some of this information as well as more in the talk, Deracializing Medicine, the Role for Pathology and Pathologists. And down the road a piece, the National Academies of Sciences, Engineering, and Medicine has convened another committee to look at the use of race and ethnicity in biomedical research. So for those of you who feel like genetics and genomics isn't really your focus, there will be something else that will be useful somewhere down the road. Now, this was a very challenging talk for me to put together. I am neither a geneticist nor a genomicist, and I did quite frequently feel like a dog trying to comprehend quantum mechanics. Now, I did have the help of two scientists who do not look like the Gary Larson depiction. They are these two here, Dr. Joseph Graves Jr., who's at North Carolina A&T State University and is a frequent collaborator with me, and Dr. Charmaine Royal, who, as I mentioned, is one of the co-chairs of this study. Here is a list of my references, and I will take any questions.